Hello, my name is Dr. Cesar Rodriguez-Saona. I'm the blueberry entomologist at Rutgers University. Here is my email in case you need to contact me, crodriguez@njas.rutgers.edu. Um, Today's lecture is going to be about blueberry IPM, or integrated pest management. Blueberries are an important crop in the United States. It ranks second among the most important commercial berries and has a value of about $1 billion. It is now produced worldwide, and it is consumed mainly due to its known health benefits since it contains high levels of antioxidants. Cultivation of blueberries started in the United States, and now they are produced worldwide. Production has increased in other countries, including Mexico, many countries in South America, in Europe, Africa, Asia, and in Australia. Production of blueberries is expected to increase worldwide due to increasing demand, particularly in South America, Europe, and in Asia. In the United States, high bush blueberries are produced in the eastern half of the country and also in several western states. Most of high bush blueberries are produced in Washington, Oregon, Michigan, Georgia, California, and New Jersey. New Jersey ranks, depending on the year, sixth in blueberry production in the country. The major producer of low bush or wild blueberries is Maine. Here, I will just discuss high bush blueberries that are the only species of blueberries that are grown in New Jersey. New Jersey is the highest producer of high bush blueberries in the Northeast region of our country. This is an important fruit crop in our state that is grown in over 9,000 acres. Most of the production is sold as fresh fruit, and this is a crop that relies on pollinators, including commercial honeybees for pollination. Most of the blueberry production in New Jersey takes place in an area known as the Pinelands or Pine Barrens. This is a national reserve located in the southern part of the state. It is a beautiful region of the state that is characterized by pine forests, rivers, and lakes, and provides places for hiking, canoeing, and kayaking. An important fact is that blueberries were first domesticated in New Jersey. These efforts started about 100 years ago. For this reason, a research center was established in Whitesburg for the study of blueberries in 1918. This center was then moved to Pemberton, and in 1962 to its new facilities in Chatsworth, where it is currently located. Two important entomologists that worked at the Rutgers Marucci Center are P. Marucci, Philip Marucci, after whom the center was named, and my predecessor, Dr. Sridhar Polaparapu. Several issues have caused changes in blueberry pest management practices in recent years. These include the fact that blueberries are a specialty crop grown in limited acreages in the US, the passing of the EPA Food Quality Protection Act that restricts use of certain broad-spectrum insecticides, maximum residue limits that are critical for export, and a critical issue is the arrival of invasive pests that have disrupted existing IPM programs. In a recent publication, we search the literature for all the papers on blueberry pest management since blueberries were domesticated around 1914. The number of studies have increased in the past 50 years, as you can see in this figure, likely due to the increase in production nationwide and worldwide. Throughout these past 50 years, the number of studies on monitoring have increased, as you can see by the red line. Also, the number of cultural controls were high in early years of production and have increased again in the past 30 years. Chemical control has been critical in blueberry pest management, and this is reflected by the number of studies throughout the years, which have remained constantly high. There has been also interest in biological control since the beginning of commercial blueberry production, but methods to increase biological control have been tested only recently. Also, research on behavioral control and host plant resistance have only increased in the past 30 years. This graph summarizes all the studies across different IPM tactics. Clearly, most research have been done on monitoring and in chemical and biological control, and a lot less on behavioral control and in host plant resistance. Most of the studies came from the United States and Canada, and within the United States, they came from the major blueberry producing states, New Jersey, Michigan, and Maine. The most studied insects include the blueberry maggot fly, the spotted wing Drosophila, cranberry fruitworm, and thrips, all of them direct pests that can damage the fruit and reduce yields. Aphids and sharp-nosed leafhoppers, two insect vectors of diseases, have also received great attention. Blueberries in New Jersey are susceptible to at least 17 insect pests throughout the season. So this chart shows when growers should monitor for these pests in gray, and in black when control treatments should be applied if needed. In New Jersey and other northern states, blueberry passed the winters in a dormant state. During this period, the only target for pest control are scales. There are several species of scale insects that attack blueberries, with Putnam scale being one of the most important. This insect feeds on plant sap, reducing plant vigor and yield. Control efforts should target the immature first instar nymphs, known as crawlers, as well as the overwintering adult stage. Cultural control practices include pruning and removing infested canes. And chemical control options include horticultural oils and insect growth regulators. During the bud development and prior to bloom, the primary target pest is cranberry weevil. Cranberry weevils, also known as blueberry blossom weevils, are brown beetles with white markings on the elytra. They have a short snout and overwinter as adults in wooded areas or unkept fields. Because adults drop off the bushes when disturbed, a beating tray is used to monitor their populations. The action threshold is 5 weevils per bush, or at least 20% injured clusters. Several pyrethroid insecticides are effective for their control. Several lepidopteran pests may be active during bloom, including leaf rollers, spanworms, and spongy moth larvae. Several species of leaf rollers feed on blueberry fruit and foliage, including oblique-banded leaf roller and red-banded leaf roller. These closely related species belong to the family Tortricidae, and they overwinter as pupae or mature larvae and have multiple generations per year. Larvae construct shelters using leaves and silk, feeding on buds in the spring and later on leaves and fruit surfaces. The adults can be monitored using pheromone-baited traps, and for the control, we recommend insect growth regulators and other reduced risk insecticides. There are also options that are available for organic production. Spongy moth is an invasive forest pest characterized by periodic population outbreaks that occur every 10 years, in about every 10 years, and may last 2 to 3 years. The larvae can be transported by wind from trees into blueberry fields, a process known as ballooning, and are active during the bloom period. Spanworms or inchworms are recognized by their slender bodies and distinct looping movement due to the presence of legs at the front and rear of the body. They feed on young buds, causing characteristic holes. So most insects that are pests in blueberries are active soon after bloom. These include plum curculio, cranberry fruitworm, thrips, aphids, and oriental beetle. Plum curculio is a weevil that overwinters as an adult, primarily in wooded areas near blueberry fields. It completes a single generation in blueberries. The adults feed on young fruit, causing characteristic feeding scars, while the females lay eggs in the fruit and create crescent-shaped oviposition scars. The infested fruit often turn purple and drop from the bush. The mature larvae exit the fruit to pupate in the soil, and the next generation of adults emerge in July and August. There are several insecticides that are registered for managing plum curculio. Cranberry fruitworm is an occasional pest of blueberries in New Jersey. This insect overwinters as a mature larva inside a hibernaculum, and adults emerge during bloom. After mating, females lay eggs singly in the calyx cap of green fruit. The larvae then emerge and bore into the fruit at the stem end. They may web together several fruits, and frass is often visible outside the fruit. The adults can be monitored using pheromone traps, and insect growth regulators and reduced risk insecticides can be used to manage this pest. Here you can see the eggs in the calyx cap of green fruit, and also you can see a larva boring a hole at the stem end. Older larvae may feed on multiple fruit, creating webs and visible frass. Aphids are a sporadic but significant pest of blueberries, particularly because they can transmit an important virus that causes scorch disease. These insects overwinter as eggs on the bushes and hatch in the spring. The young aphids feed on new foliage, and populations increase through asexual reproduction, parthenogenic reproduction during June and July. When the colonies become crowded, winged aphids are produced, allowing them to disperse. In the fall, the males are produced and they mate with the females, and the females lay their eggs. Systemic insecticides can be effective for managing aphid populations. Because aphids are vectors of viruses, there is little tolerance for their presence. However, insecticide use can also harm their natural enemies. Aphids feed on the underside of leaves and on young foliage at the bottom of bushes, making them difficult to manage. For this reason, thorough insecticide coverage is critical for effective control. A pest of concern is flower thrips. Several species feed on blueberries, with some targeting leaves and others flowers. Thrips can be active during bloom, but in New Jersey, their populations often peak after bloom. They are found feeding on the petals, causing petal discoloration, as well as curling and malformation of the leaves. Another pest of concern is the gallmidge, also known as the cranberry tipworm. Females lay eggs on developing tissues, causing the death of growing tips. Gall midges have short generation times and can complete several generations per year, and they overwinter in the larval stage. The most important soil-feeding insect pest of blueberries in New Jersey is the oriental beetle. This is an invasive pest that is widely distributed across the US and Canada. It can have a particularly severe impact on young plantings by damaging the root system. The adults can be monitored using pheromone traps. Two fruit-feeding insects that attack blueberries during the harvest are the native blueberry maggot and the invasive spotted wing Drosophila. Blueberry maggot fly overwinters in the pupal stage, and the adults emerge in early June, coinciding with the start of harvest. Females required about 10 days of feeding to become reproductively mature. This is considered a quarantine pest and is regulated when exporting blueberries to other countries. Spotted wing Drosophila is an invasive pest from Asia. It was first detected in the continental USA in 2008 in California and in New Jersey in 2011. It feeds on multiple hosts, including strawberries, raspberries, cherries, and blueberries. And unlike other Drosophila species, females can lay eggs in ripening fruit because they have a prominent serrated ovipositor. Since the invasion of this pest, it has become the primary threat to blueberries, driving insecticide applications and largely replacing the native blueberry maggot. The only target pest in New Jersey during post-harvest is the sharp-nosed leafhopper. Sharp-nosed leafhoppers transmit a phytoplasma that causes stunt disease. This insect completes 2 generations per year and overwinters as eggs. The eggs hatch in mid-May, and the adults of the first generation emerge in June. The second generation adults appear in late August through early September. Our current IPM program at Rockers is based on weekly monitoring of these major pests and the applications of insecticides if populations reach a threshold or are above a state mean. Most of the insecticide applications target pests that directly attack the fruit and are thus used after petal fall. The IPM program is coordinated by Janine Spies and has about 40 subscribed growers. The information on pest management is provided to our growers in a weekly basis through newsletter articles during the field season. Management decisions are based on monitoring and existing thresholds set for different periods: before bloom, during bloom, and after bloom. However, thresholds are available only for a few insect pests. These include the cranberry weevil, thrips, and blueberry maggot. For other pests, trap counts are used to help determine the timing of insecticide applications, such as for cranberry fruitworm, oriental beetle, and Spadawin Drosophila. There are several constraints that limit wider adoption of IPM among blueberry growers. First, there is low tolerance for infested fruit. We have also quarantine pests that restrict exportation of fruits to other countries. We have insects that vector diseases such as aphids and sharp-nosed leafhoppers. And there is constant risk of new invasive pests. The fresh market requires high quality of fruit, and we have only few reliable action thresholds. So what can we do about this? We need to improve our monitoring tools to develop more accurate action thresholds. For these, we're testing new monitoring tools for cranberry weevil, plum curculio, and Spadawin Drosophila. We also need to evaluate new softer insecticides and implement additional IPM-based tactics, such as behavioral and biological control. We're currently testing behavioral-based control strategies for Spadawin Drosophila and also implementing biological control for Spadawin Drosophila, plum curculio, and oriental beetle through the use of entomopathogenic nematodes. To do this, my lab is using insect chemical ecology to develop new and better monitoring tools and behavior-based control strategies. We can manipulate insect behavior through the use of insect pheromones. These chemicals are involved in intraspecific communication, meaning that they trigger a response in members of the same species. There are several types of pheromones, including sex pheromones, aggregation pheromones, trail pheromones, alarm pheromones, and marking pheromones. In addition, insects use chemicals from plants, such as volatiles, that can affect their performance and preference, and thus they are involved in inter-specific communication. One such class of chemicals are volatiles that are emitted by plants damaged by herbivores and can be used to attract natural enemies to find their host or prey. In particular, my lab has been studying the effects of crop domestication on defenses against insect herbivores. Domestication of crops has altered many traits in plants, including the size, and humans have often selected for bigger reproductive organs. This group of traits that appear in the domesticated plants are known as the domestication syndrome and was referred by Darwin as a form of selection. For example, maize has been domesticated for about 9,000 years, mainly for bigger size and reproductive structures. This selection has made plants more susceptible to herbivore attack. This higher susceptibility of domesticated plants to herbivores is thought to be because of a trade-off between growth, reproduction, and defense. In fact, domestication often makes plants less resistant to herbivores by lowering the amount of secondary metabolites that serve as defenses. This results in insects performing better on crops, domesticated crops. However, these interactions have been reported in only a few crops. High bush blueberries are a perfect crop to study domestication syndromes. This crop grows next to its wild ancestors in the pinelands and has only been domesticated for about 100 years. Wild and cultivated blueberries differ in many traits, but mainly in size of the bush, size of the fruit, and the number of fruit per bush. So we studied the consequences of blueberry domestication on the invasive pest Sparrowing Drosophila. Our prediction was that domesticated fruit would be more susceptible than wild fruit to this pest. So we hypothesized that domesticated blueberries are more susceptible to an invasive insect frugivore than the wild blueberries. To test this hypothesis, we collected fruit samples from 12 sites across the pinelands. These sites ranged in management practices from low to high inputs. Fruit was taken to the lab, placed in cups, and then exposed to flies in single choice essays as shown in these pictures. We also we measured the number of eggs that were laid per fruit and also incubated those berries to determine adult emergence. Here you can see the eggs with their breeding tubes. And this is how we counted the number of eggs per fruit. And then we incubated the fruit and measured adult emergence. We found more eggs laid in domesticated than wild fruit in both years of the study. So we found that domesticated blueberries had about twice the number of eggs compared to wild blueberries. We also found higher emergence of adults from cultivated fruit in both years. Domesticated blueberries had also about twice the number of adults emerging from the fruits compared to wild blueberries. And the percent of larval survival was not significantly different in the first year, but was lower in the wild berries in the second year. So survival of the larvae was higher in domesticated blueberries than in wild blueberries, at least in one of the years of the study. So we also conducted dual choice tests where flies were given a choice between wild and cultivated fruit. So we did this in the arenas shown in this figure, in these pictures, inside cages. We found that females prefer to lay eggs in cultivated than in wild berries when given a choice. So domesticated blueberries were more preferred for oviposition than wild berries. So we measured several physiological and chemical traits in these fruits. We predicted based on previous studies that firmer fruit will result in lower infestation and that higher sugar content will increase infestation. So as expected, domesticated fruit were bigger, but also firmer than the wild blueberries. So domesticated blueberries are larger and firmer than wild berries. Wild berries had lower pH, thus were more acidic, but had higher levels of sugar than the cultivated berries. So domesticated blueberries are less sour, but less sweet than wild blueberries. Finally, the wild blueberries had higher concentrations of defensive compounds, such as phenolics and anthocyanins. So domesticated blueberries are less defended and also lighter in color compared to wild blueberries. These anthocyanins provide the blue color to the blueberries. So in conclusion, we found that domesticated blueberries are more susceptible to this invasive frugivorous pest, the Spotted-winged Drosophila. We found that females of Spotted-winged Drosophila are choosy, but choice was not associated with fruit sweetness or firmness. We found a correlation between preference and performance with fruit size, pH, and defense levels of phenolics and anthocyanins. So there are multiple plant traits that need to be measured to better understand the effects of domestication on herbivores such as Spotted-winged Drosophila. I invite you to visit my website to learn more about our program. This site that I'm showing you is linked through the Rutgers B. Marucci Blueberry and Cranberry Research and Extension Center. I also have a personal website, and you're welcome also to contact me if you have any questions. Thank you.